copper 2 sulfate solution. This will result in a violet or purple color, which also indicates the presence of protein. Then the xanthoproteic test. To a small amount of protein solution, you add small amount of concentrated triazonitrate 5 acid and concentrated amino solution in excess. The mixture, you gently warm it and later allow it to cool. This will result in a red or orange precipitate during warming. Then on cooling, it will change to a deep yellow color, which indicates the presence of protein. Now, we move on to test for fats and oil. The first test here, we have also 3 tests. We have the translucent white paper test, the Sudan 3 solution test, then the osmic acid test. This translucent white paper test, you put a drop of oil on a piece of white paper, then you hold the piece of paper against a light source. This one you can do just immediately after this class. Put a drop of, let's say, your granite oil or palm oil on a white paper. Then you take the paper close to a source of light. Then you discover that the translucent part will show, you discover that it will be translucent. That is, it will kind of transparent. You find the oily part, or like the part that you did not put oil, it will be kind of transparent. You can see through it. So the translucent part shows the presence of fat and oil. Then the next test for fat and oil, you have the Sudan 3 solution test. Add a few drops of Sudan 3 solution to oil in a test tube. Then you boil the solution. A red color shows the presence of oils or fats. Then the third test is osmic acid test. Add a small quantity of osmic acid test to equal volume of oil, the fat and oil in the test tube, that is equal volume of osmic test to fat and oil in the test tube. Then you boil the mixture. You discover that you find a black precipitate, which also indicates the presence of fat and oil. Now, test for water. That's the last food test that we'll be talking about. You dip a blue dry cobalt chloride paper. That's a kind of paper. A blue dry cobalt chloride paper in a food item. When you dip it in a food item, the paper changes. You know, it's a blue colored paper. It changes from blue to pink, and that indicates the presence of water. Now we'll be talking about the modes of nutrition, various modes of nutrition. Earlier we talked about autotrophic and heterotrophic nutrition. So we'll now talk more about that. And under autotrophic nutrition, I said autotrophic nutrition involves the manufacture of food by some organisms on their own. They manufacture their food in the presence of sunlight by an organelle called chloroplast in them. So we talk about chemosynthetic nutrition. Certain bacteria are able to synthesize organic compounds from simple organic compounds such as carbon 4 oxide, water, hydrogen sulfide, ammonia to provide their food. Certain bacteria are able to synthesize organic compound from simple inorganic compound. That's a kind of autotrophic nutrition done by the bacteria. Also, the energy used for the synthesis comes from the oxidation of the inorganic materials. Example, we have sulfur bacteria such as Nitrosomonas and Nitrobacter. Those are examples of sulfur bacteria. They convert ammonia to nitrites, while Nitrobacter converts nitrites to nitrates. So Nitrosomonas converts ammonia to nitrites, while Nitrobacter converts nitrites to nitrates. Then to heterotrophic nutrition, I said some organisms, they eat various kinds of food. They do not manufacture their food on their own. So we have holozoic nutrition. This involves feeding on green plant products and other organisms. That is feeding on green plant products, feeding on plants, and we have grouped them into, they are found in carnivores, herbivores, omnivores, and scavengers. Carnivores are flesh eaters, lion, tiger. Herbivores, plant eaters, dog, we have sheep, we have goats, we have cattle. They feed only on plants. Then we have omnivores, that is, they feed both on plants and on flesh. Example, we have pig, we have man. Then we have scavengers. Scavengers are organisms that feed on dead animals, and examples of such, we have the vulture. Vulture is a scavenger. Then we also have parasitic nutrition under heterotrophic nutrition. This involves an organism feeding on another organism in order to derive nutrients, but it does the feeding on another organism to the detriment of that organism. And examples of organisms that undergo parasitic nutrition, we have tapeworm, Taenia sonia, we have Plasmodium that causes malaria, we have roundworm, we have liver flukes, we have ticks, as we can find on the body of dog, fleas, bugs, aphids, guinea worm, hookworm, and all animal parasites. They undergo parasitic nutrition. Then we also have plant parasites. Plant parasites include doda plants, that's Casita filiformis. Then we have the mosquito, that is another parasitic plant. Animal parasites are classified into two: endoparasites and ectoparasites. That is, parasites that are found inside the organism, the prey. That's the endoparasites. Then ectoparasites that you find on the body of the host. Ectoparasites. Examples, we have flea. We find fleas on the body of some animals. For instance, we find fleas on dogs. Then we have bed bug, you have tick. Those are examples of ectoparasites. You even find it in humans. On the head of some children, you find fleas on their head. So that's an example of ectoparasites. Even on the body, maybe when they have interacted, played on sand, and those things they've got infected, so it's on the body. That's ectoparasite. Then another group is the endoparasite. It lives in the body of the host. And examples, we have tapeworm, like I mentioned earlier. It is associated with pigs. Tapeworm is associated with pigs, which is the secondary host. And you also find it in man. Man is the primary host. Man gets it from pig. When you eat pork, that is meat from pig, that are not well cooked, you can get taenia, that's tapeworm, into your system by eating such meat. When it's not well cooked, you can get tapeworm. So that's why you have to be careful to make sure what you eat is well cooked. Then tapeworm body is flat, which provides large surface area for the absorption of digested food from the host. It has flat body, so it can easily absorb digested food from the host. It's found in the abdomen of the host. Also, another example of endoparasite is the liver fluke. It is commonly found in the liver of sheep. The next endoparasite is that we will be talking about is the filaria worm. That's the Wuchereria bancrofti. It causes elephantiasis, the swelling of the legs, and it is associated with goats, cattle, and sheep. So they are the ones that carry it, and you find it in man too. Especially, they get it in soil and in water areas. Then you have another endoparasite. We have the roundworm. That's the Ascaris lumbricoides. It is commonly found in the alimentary canal of pig too. You find it in pig. So Ascaris lumbricoides, that's another endoparasite. Also, we have guinea worm. Guinea worm is also another example of endoparasite. That's the Drancoculus medinensis. Also, we have parasites in plants. First, we have the Casita filiformis. That's the doda plants. It's a complete parasite because it's absorbed already made food from its host. This particular plant, the casita plant or the doda plant, has no roots, no chlorophyll, so it cannot manufacture its own food. It only depends on the host. Also, another example of parasitic plant is mistletoe. It's a partial parasite. It has roots only to penetrate the xylem of its host to absorb water for the process of photosynthesis. This particular mistletoe has green leaves, so it can manufacture its food. Though it's a partial parasite, it uses its roots to absorb water from the xylem of its host. Also, another mode of nutrition that we'll be talking about is a saprophytic mode of nutrition, and this involves organisms feeding on dead and decaying organic matter. The saprophytes secrete digestive enzymes on the substrate, that is on the dead organic matter which they grow. So the enzymes are secreted to digest the substrate externally, and the food is absorbed into the body of the organism. Examples of saprophytic organisms: we have rhizopus, we have mushroom, we have mucor, we have penicillin, and toadstool. Another mode of nutrition, we have the symbiotic nutrition. Symbiotic nutrition involves two organisms of different species that live together and derive food from each other. They both derive food from each other. They are organisms of different species. They come together, they derive food from each other. They both gain. None of them is losing. They both gain from each other, and they develop protection, shelter, and reproduction from the association. Not only food, they get fed, they derive protection, they derive shelter and reproduction from the association. Examples of organisms that undergo symbiotic nutrition: we have the nitrogen fixing bacteria and the root nodules of leguminous plants. The root nodules of leguminous plants and nitrogen fixing bacteria. The root nodules, the bacteria helps